Hi students, we are going to learn about structure of proteins. So there are primary structure, secondary structure, tertiary structure and quaternary structure. So as you know, proteins they are biological molecules and which are formed by the polymerization of amino acids. So proteins are polymers of amino acids. Many amino acids are linked together by many bonds. Mainly the bond is peptide bond. But some other secondary bonds are also helps the formation of the many protein structure that are disulfide bridges, hydrogen bonds, ionic bonds, hydrophobic bonds, etc. As you know, hydrophobic force and the relationship between water and hydrophobes. What do you mean by hydrophobes? Low water soluble substances. And ionic bond formed from the electrostatic attraction between oppositely charged ions, that ionic bond. Then disulfide bridges, you know, cysteine ennu parayunna oru amino acid undu. Mainly 20 amino acids are there. Among that one amino acid, the name is cysteine. So the bond is formed between two cysteine molecules, that is disulfide bond. How is it formed? The thiol SH groups of two cysteine molecules that may be oxidized to a cysteine. So a bond or linkage is formed between these two. That bond is known as disulfide bond. So proteins, they are polymers formed by the polymerization of its micromolecules that are amino acids. Pala amino acids join ചെയ്താണ് protein ഉണ്ടാവുന്നത്. അപ്പോൾ ഇങ്ങനെ ഈ അമിനോ ആസിഡിനെ ഒരു ഇങ്ങനെ ജോയിനിങ്ങിന് ഹെൽപ്പ് ചെയ്യുന്ന മെയിൻ ആയിട്ടുള്ള ബോണ്ട് പെപ്റ്റൈഡ് ബോണ്ടാണ്. But in addition to this peptide bond, many other secondary bonds are also present, which are the disulfide bridges, hydrogen bonds, ionic bond, hydrophobic bonds, etc. So a peptide that is formed of 2 to 10 amino acids. 2 to 10 amino acids. അത്രയും amino acids മാത്രം join together ചെയ്തുണ്ടാവുന്ന peptide ആണ് oligopeptide. Oligopeptide. And peptide with more amino acids, they are known as polypeptides. Peptide with more amino acids, they are known as polypeptides. So proteins, they are having two ends. One end is having amino group or NH2 group and the other end is having carboxylic group or COH group. So the end which is having amino group or NH2 group, that is known as N-terminal end, and the other end which is having carboxylic group or COH group, that are known as C-terminal ends. Okay. So how does a peptide bond is formed? As I told you, the peptide bond is formed between two amino acids. This formed between amino group of one amino acid and carboxylic group of the another amino acid. How does this formed? So here this is one amino acid. So this is NH2 amino group or amino terminal. This is carbon, here hydrogen, here also this one side is alkyl group or R group, that is varying, but here that is H, it's one amino acid, and this N, that is carboxylic group COH, and this is another amino acid. So here you can see two amino acid. This is one amino acid and this is one amino acid. ഇവിടെ നോക്കിയേ, the OH group of carboxylic group of first amino acid reacts with hydrogen of NH2 group of the second amino acid. This is first amino acid, this is second amino acid. This first amino acid in the carboxylic group in the OH, second amino acid in the amino group in the H join one water molecule is releasing. H plus OH, H2O, അത് release ചെയ്തു. So what all are remaining here? So this here you can see ഇവിടെ വരെ സെയിം ആണ്. ഇവിടെ നോക്കുക COHൻ്റെ CO മാത്രമേ ഉള്ളൂ, NH2ൻ്റെ NH ഉള്ളൂ. COയുടെ ഈ OHും NH2ൻ്റെ ഈ Hും കൂടി ജോയിൻ ചെയ്ത് വാട്ടർ മോളിക്യൂളായിട്ട് റിലീസ് ചെയ്തു. So here you can see CO-NH bond, that is peptide bond. So a peptide bond is results from the elimination of water molecule from the OH group of the carboxylic group of first amino acid and hydrogen of the amino group of the next amino acid. So in this way that peptide bond is forming. This peptide bond is the major bond present in a protein. ഏറ്റവും ഇമ്പോർട്ടൻ്റ് ആയിട്ടുള്ള ബോണ്ട് എന്ന് പറയുന്നത് ദിസ് പെപ്റ്റൈഡ് ബോണ്ടാണ്. ഇൻ അഡീഷൻ ടു ദിസ് പെപ്റ്റൈഡ് ബോണ്ട് എ വെറൈറ്റി ഓഫ് സെക്കൻഡറി ബോണ്ട്സ് ആർ ഓൾസോ പ്രസൻറ്റ്. ആസ് വി ഡിസ്കസ്ഡ് ദേ ആർ disulfide bridges, hydrogen bonds, ionic bonds, hydrophobic bonds, etc. But this is important. How does a peptide bond is formed? A peptide bond results from the elimination of a water molecule from the OH of carboxylic group of first amino acid and hydrogen of the amino group of the next amino acid. This is peptide bond. And then based on the degree of complexity, 4 basic structural levels of organization of protein structures have been recognized, which are the primary structure, secondary structure, tertiary structure and quaternary structure. And when Linus Pauling, that he was awarded Nobel Prize for explaining these different levels of protein structure. So based on the degree of complexity, we are dividing the protein structure into 4: primary structure, secondary structure, tertiary structure and quaternary structure. And what is primary structure? Actually, നമ്മൾ ഈ സ്ലൈഡിൽ കണ്ടതുപോലെ here you can see one amino acid, second amino acid. അപ്പോൾ ഇവിടെ ഇവർ തമ്മിൽ ഇവിടെ ഒരു പെപ്റ്റൈഡ് ബോണ്ട് കൊണ്ട് ജോയിൻ്റ് ആയി. ഫസ്റ്റ് അമിനോ ആസിഡും സെക്കൻഡ് അമിനോ ആസിഡും തമ്മിൽ പെപ്റ്റൈഡ് ബോണ്ട് കൊണ്ട് ജോയിൻ്റ് ആയി. So like that here third amino acid will come, fourth amino acid will come, fifth amino acid will come. So between every amino acid we can see this peptide bond. So this type of linear arrangement of amino acids by peptide bond, that is known as primary structure. So it defines the exact sequence of amino acids, and we can count the total number of amino acids from the primary structure, and we can note down the variety of amino acids, and we can discover the order of amino acids in the primary structure. And in the linear polypeptide, amino acid at one end contains free amino group and amino acid at the opposite end carries free carboxylic group. So those amino acids which containing free amino group is called N-terminal or amino-terminal amino acid, and those with the free carboxylic group is called C-terminal or carboxy-terminal amino acid. So while explaining the primary structure, the N-terminal amino acid is written in the beginning and C-terminal amino acid in the end. Why? എന്തുകൊണ്ട് അങ്ങനെ എഴുതുന്നത്? എപ്പോഴും നമ്മൾ പ്രൈമറി സ്ട്രക്ചർ എഴുതുമ്പം N-terminal at the beginning and C-terminal at the end. Why? Because protein synthesis occurs in N to C-terminal direction. Okay, so see this picture. Here you can see primary structure of the protein. So here this is the structure of an amino acid. So carbon, hydrogen, R group, that is a side chain alkyl group, that is varying. Here COH, NH2. ഈ ആർ ഗ്രൂപ്പ് എന്ന് പറയുന്നത് മാത്രമാണ് വേരി ചെയ്ത് വരുന്നത്. ബാക്കി എല്ലാം സെയിം ആയിരിക്കും അമിനോ ആസിഡിന്, എല്ലാ അമിനോ ആസിഡിലും. സോ ദിസ് ഈസ് NH2 ഗ്രൂപ്പ്, NH2 ഗ്രൂപ്പ്, ദിസ് ഈസ് COH ഗ്രൂപ്പ്. അപ്പോൾ ഇങ്ങനെയുള്ള പല അമിനോ ആസിഡുകൾ 1 by 1 ആയിട്ടിരിക്കുന്നതാണ് പ്രൈമറി സ്ട്രക്ചർ നമ്മൾ പറഞ്ഞു. സോ ദിസ് ഈസ് 1 amino acid, second amino acid, third amino acid, fourth amino acids and so on. In between every amino acid you can see a peptide bond also. Okay. So here you can see this is the first amino acid. The first end is beginning തുടങ്ങിയിരിക്കുന്നത് ഏതെന്താണ്? NH2 or amino group. This is amino terminal. beginning of the primary structure, that's with the N-terminal end. This is free. This is bond involved. This is a peptide bond form. But first amino acid in the carboxylic group is involved in that peptide bond formation with the second amino acid. So this is second amino acid. The second amino acid in the amino group is NH. First amino acid in the carboxylic group is OH join the CO NH bond peptide bond undi water molecule release like that. Second molecule in the carboxylic group and amino group of the third amino acid is joining and form the peptide bond. And fourth amino acid, you just look here, in the case of fourth amino acid, this is primary structure consists of only 4 amino acids. So here the beginning or first end is free is amino terminal because it has free NH2 group. Like that, the last amino acid, the last amino acid in the amino group മാത്രമാണ് peptide bond involved ആയിരിക്കുന്നത്. 3ഉം 4ഉം amino acid തമ്മിൽ peptide bond ഉണ്ടാവുന്നതിൽ fourth-ന്റെ amino group ആണ് involved ആയിരിക്കുന്നത്. Then fourth amino acid is the last amino acid in this structure. So the carboxylic group is free. ഇവിടെ COH ഉണ്ട്. ഈ carboxylic group free ആണ്. കാരണം ഇത് കഴിഞ്ഞ ഒരു amino acid ഇല്ല അവിടെ peptide bond form ചെയ്യാൻ. So the last amino acid that has free carboxylic group, so it is C-terminal end. So ഇത് 4 amino acid ഉള്ളതാണ്. ഇങ്ങനെ എത്ര amino acids ഉണ്ടെങ്കിലും നമ്മളിങ്ങനെ ലീനിയർ ആയിട്ട് എഴുതിയാൽ ഫസ്റ്റ് അമിനോ ആസിഡിന് ഫ്രീ NH2 ഗ്രൂപ്പും ലാസ്റ്റ് അമിനോ ആസിഡിന് ഫ്രീ കാർബോക്സിലിക് ഗ്രൂപ്പും ഉണ്ട്. സോ ബിഗിനിങ് N ടെർമിനൽ എന്നും എൻഡിങ്ങിൽ നമ്മൾ C ടെർമിനൽ എന്നുമാണ് പറയുക. ബിക്കോസ് പ്രോട്ടീൻ സിന്തസിസ് occurs in N to C direction. So these are amino acids. These are various amino acids linked together by peptide bond. So the name the bond involved in primary structure of protein only peptide bond. Okay. And the next is secondary structure. The secondary structure means the elongated polypeptide chain that undergoes folding or coiling in a regular manner. Then we get secondary structure. ഈ ഇരിക്കുന്ന നീളത്തിൽ ലീനിയർ ആയിട്ടിരിക്കുന്ന ഈ പ്രൈമറി സ്ട്രക്ചറുള്ള പ്രോട്ടീൻ ദാറ്റ് അണ്ടർഗോസ് റെഗുലർ ഫോൾഡിംഗ് ഓർ കോയിലിംഗ്. സോ ദെൻ വി വിൽ ഗെറ്റ് സെക്കൻഡറി സ്ട്രക്ചർ. ആൻഡ് ഇൻ അഡീഷൻ ടു പെപ്റ്റൈഡ് ബോണ്ട്, hydrogen bonds are also be present in the secondary structure. Mainly there are 2 common secondary structures which are the right-handed alpha helix and beta pleated sheets. Then alpha helix, it is formed by the right-handed coiling of a single polypeptide chain. ഒരു സിംഗിൾ പോളിപെപ്റ്റൈഡ് ചെയിൻ ആയിരിക്കും, ഒരൊറ്റ പോളിപെപ്റ്റൈഡ് ചെയിൻ റൈറ്റ് ഹാൻഡഡ് സൈഡിൽ കോയിൽ ചെയ്താലാണ് ആൽഫ ഹെലിക്സ് ഉണ്ടാകുന്നത്. ഇറ്റ് ഈസ് സ്റ്റെബിലൈസ്ഡ് ബൈ ഹൈഡ്രജൻ ബോണ്ട്സ് ഓൾസോ. The whole chain is thus hydrogen bonded at every 4th amino acid residues. So amino acid placed at 3 or 4 apart in the linear chain is specially brought close together. Why? The amino acid, the hydrogen bonds are at every 4th amino acid residues. So in addition to peptide bond, we can see hydrogen bonds also in the secondary structure. Alpha helix is also a secondary structure, is a type of secondary structure. So we can see hydrogen bonds in the alpha helix. In the case of alpha helix, the hydrogen, the whole chain is hydrogen bonded at every 4th amino acid residue. So the amino acid placed at 3 or 4 apart in the linear chain is specially brought close together. So this is the figure of this alpha helix. So right-handed coiling of the linear polypeptide chain. So this is C end and this is N terminal end. Okay. And the next is beta pleated structure. It is formed by the folding of polypeptide chain, but folding in another manner. Here many polypeptide chains folded in a regular manner come to lie side by side. So the amino group of one chain, so look on this diagram. Sorry, the amino group of one chain faces the carbonyl group of the next chain. So the structure is stabilized by hydrogen bonds between adjacent chains. So example of beta pleated sheet, that is silk protein. Which is the silk protein? Fibroin. This is the beta pleated sheet. This is also a type of secondary structure. Next structure, level of structural organization, that is tertiary structure. In the tertiary structure, long polypeptide chains undergo folding and coiling upon themselves like a hollow woolen ball. Woolen ball pole ഒരൊറ്റ polypeptide തന്നെ ഒരു long ആയിട്ടുള്ള polypeptide chain undergoes folding and coiling upon themselves. Single polypeptide chain, but folding upon themselves and compress itself into a globular form. The globular shape. That's why I have told you it's like a hollow woolen ball, a ball like structure. A long polypeptide chain coiling and folding upon themselves. നീളത്തിലുള്ള ഒരു polypeptide chain അത് തന്നെത്താൻ അതിൽ തന്നെ ചുറ്റി പിണഞ്ഞു കിടക്കുന്നു, like a hollow woolen ball, and it compresses itself into a globular form. That is tertiary structure. Example of tertiary structure, that is myoglobin. This globular proteins, if the proteins are very compact, they are called globular proteins, and if less compact, they are called fibrous protein. But tertiary structure എന്താണ് പഠിക്കുക? Then tertiary structure is stabilized by 4 types of bonds. Which are they? Disulfide bonds between two cysteine molecules. Cysteine molecule thiol group ആണ് involve ചെയ്യുന്നത്. But bond അത് oxidize ചെയ്ത് കഴിയുമ്പം cysteine becomes cysteine. So disulfide bond between two cysteine molecules. Then hydrogen bonds, bonds between acidic amino acids and basic amino acids, ionic bonds. Then hydrophobic bonds. So primary structure, only peptide bond is involving. Secondary structure, peptide bond and hydrogen bonds are involving. In the tertiary structure, 4 types of bonds at present, which are the disulfide bond between cysteine molecules, hydrogen bonds, hydrophobic bonds, and bonds between acidic and basic amino acids. And this tertiary structure that offers definite structure and functional properties like enzyme activity to the proteins. Proteins, some proteins are functioned as enzymes. So enzyme ആയിട്ടൊക്കെ proteins act ചെയ്യുന്നത് ആ tertiary structure attain ചെയ്യുമ്പോഴായിരിക്കും. So the tertiary structure offers definite structure and functional properties like enzyme activity. And the next, it gives 3-dimensional view of the proteins. 3-dimensional view of the proteins. And the another feature is the bonds present in tertiary structure are weak and fewer in number and can be easily disrupted by agents like heat, acidity, etc. So this disruption of bonds that changes the folding and damages the properties of protein, that is known as denaturation. നമ്മൾ എൻസൈംസിൻ്റെ ഒക്കെ കേസിൽ പറയുന്നുണ്ട് ഹൈ ടെമ്പറേച്ചർ വന്നാലൊക്കെ ഇത് ഡീനേച്ചർ ആവും, അതിൻ്റെ സ്ട്രക്ചർ ലോസ് ആവും, അതിൻ്റെ ആക്ടിവിറ്റി കുറയും അങ്ങനെ. അപ്പോൾ അത് ബിക്കോസ് വെൻ എ പ്രോട്ടീൻ ഈസ് ഇൻ ദ ടെർഷ്യറി സ്ട്രക്ചർ, the bonds present in it are weaker and are easily disrupted by other agents like heat, acidity, etc. So this disruption of bonds changes the structure of the protein and damages its properties. That process is known as denaturation. So it's a tertiary structure. And the last is quaternary structure. It defines the spatial arrangement of different polypeptide chains in a complex protein containing many polypeptides. ഇവിടെ ഒരു polypeptide അല്ല. In the tertiary structure, a long polypeptide undergoes folding and coiling upon themselves and attain a globular shape like hollow woolen ball. But here, in the quaternary structure, it defines the, actually in the quaternary structure, many polypeptides are present. It's a complex protein. It consists of many polypeptides. So these different polypeptides are undergoes folding or coiling. So quaternary structure defines the spatial arrangement of different polypeptide chains in a complex protein containing many polypeptides. So if the subunits are identical, the protein is said to be homogeneous quaternary structure. Or if the subunits are different, it is known as heterogeneous quaternary structure. Okay, it consists of many polypeptide units or subunits. If the subunits are identical, the protein is said to be homogeneous quaternary structure, and if different as in hemoglobin, quaternary structure example is hemoglobin, tertiary structure example is myoglobin, quaternary structure the example is hemoglobin. Hemoglobin, the subunits are different, so it is said to be heterogeneous quaternary structure. For example, in hemoglobin there are 4 polypeptide chains, 2 different chains, 2 alpha chains and 2 beta chains. Okay, so all the 4 globins are connected to a centrally located heme molecule to form a functional hemoglobin. So in the case of hemoglobin, we can see different subunits, 2 alpha chains and 2 beta chains. They are connected to a centrally located heme molecules. So hemoglobin attains heterogeneous quaternary structure. Okay. So it's an image of the quaternary structure, different polypeptides are present in it. So there are mainly 4 structures: primary structure, primary protein structure, a sequence of a chain of amino acids. That only. In the case of primary structure, amino acids are sequentially arranged. It's a linear form. And secondary protein structure, local folding and coiling of the polypeptide chain into either it is helix or it is sheet like structure. Fold the helix roopathil alpha helix, or it is a sheet like structure, that is beta plated sheets. And when it comes to the tertiary structure, in the tertiary structure we can see the 3-dimensional structure of the protein, because due to side chain interactions also. A single polypeptide chains undergoes regular coiling and folding upon themselves. And the last one is quaternary protein structure. This protein consists of more than one polypeptide chain. Consists of many polypeptide chain. It's a complex protein. Here also you can see it's a complex protein. Different subunits are present. Here you can see purple color, green color, light blue, violet, etc. So different subunits are present. Okay, that is quaternary structure. Thank you.